Transposons form the telomeres of Drosophila.
But what exactly are telomeres? The name telomere comes from Greek telos, meaning end, and meros, meaning part. So a telomere is a region of repetitive non-coding DNA at the end of the chromosomes, which protects the end of the chromosome from destruction. In this image you see the human chromosomes in blue with the telomeres at the end of each chromosome labeled in yellow, as the arrows indicate. The cell, however, faces a problem with the telomeres at the tip of the chromosomes. Telomeres, and thereby the chromosomes themselves, shorten because of the so-called end replication problem during DNA replication in eukaryotes. This scheme will demonstrate this problem to you. DNA replication starts within the chromosome and not on either end of it. A replication fork opens as seen in this figure. DNA polymerase, which synthesizes DNA, can only synthesize a new strand of DNA as it moves along the template strand in the 3' to 5' direction, synthesizing new DNA in the 5' to 3' direction. This works fine for the 3' to 5' leading strand of a chromosome, as the DNA polymerase can move continuously from the origin of replication until it meets another bubble of replication or the end of the chromosome. The fact that DNA polymerase cannot synthesize a new strand in the 3' to 5' direction causes a problem for the DNA replication on the so-called lagging strand. So the lagging strand is synthesized in small fragments. Each fragment being synthesized in the 5' to 3' direction, and thereby opposite the direction in which the replication fork is moving. Small molecules of RNA bind to the template strand and serve as primers for the synthesis of DNA for each of the fragments. After the RNA primers have been removed and filled with DNA bases, DNA ligase ligates the fragments together. You can see here how the lagging strand is synthesized bit by bit in small fragments that are ligated afterwards to form continuous DNA again. The single fragments are called Okazaki fragments. A chromosome has several origins of replication, where the replication forks open, as seen in the first image. Wherever the replication fork of a strand is moving towards the 3' end, the newly synthesized DNA begins as Okazaki fragments, as you can see in the second image. This continues until close to the end of the chromosome. Then, as the replication fork nears the end of the DNA, there is no longer enough template to continue forming Okazaki fragments. There is no place for another RNA primer, and DNA polymerase cannot add new nucleotides to the 5' end of the new strand. So the 5' end of each newly synthesized strand cannot be completed. Thus each of the daughter chromosomes will have a shortened telomere, indicated by the green circles. During each cell cycle the chromosomes would be shorter by up to 100 base pairs. But as usual, nature has found a solution for this problem. Telomeres consist of short terminal repeats. In humans, the sequence of these repeats is GGG-ATT, and the telomeres can be hundreds or to thousands of nucleotides long, with many of these repeats. Some of our cells, the germline cells, contain an enzyme called telomerase. This enzyme consists of a protein part, here in gray, and an RNA part, the nucleotides with the sequence AAUCCC within the GRI protein, which is complementary to the sequence of the telomeric repeats. Telomerase has reverse transcriptase activity. It can synthesize DNA by using its RNA as a template. It moves along the telomeres on the normal, not on the shortened strand, in 5' to 3' direction. It thereby elongates the telomere by adding more repeats to the chromosome's end. With the elongated strand, DNA polymerase now has enough template to synthesize the complementary DNA strand of the telomere. The mechanism of telomerase adding new telomeric repeats to the end of the chromosome counterbalances the loss of DNA through replication mechanisms. The leftover single-stranded end is probably being folded back upon itself to form a hairpin structure to prevent nucleases from breaking it down.
But what about the telomeres in Drosophila? They are different.